This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 69 for February 4th, 2010. Hello, everybody. I'm Vincent Racaniello, and it's time to talk about viruses. Joining me to do that today from Western Massachusetts is the venerable Alan Dove.
Oh, I'm venerable now. I hope I don't catch a venerable disease.
He's starting off from the start with excellent. Good to have you.
Yeah.
And that deep laugh is from down south, from Florida. It's Rich Condit.
Hello. Great to be here.
2 weeks in a row, eh?
Yeah. This is getting to be a habit.
It's a good habit to have.
Yeah, it's wonderful. I look forward to it.
It's fun, eh? Learning all about viruses.
Yeah, it's fun. Yeah, and I learn a lot.
So we have a couple of good stories as usual today. By the way, guys, Dick Despommier is back in town.
Oh, good.
Saw him yesterday. Unfortunately, he's teaching right now. And
I thought he was retired.
Yeah, I did too. I don't know why he's teaching.
These guys do this kind of stuff, you know?
Yeah, well, you have all this knowledge. It's a waste to just not use it, right?
Right.
Might as well teach. But since we weren't very flexible with the timing, he can't join us. So next time we'll have Professor Despommier back to talk about Argentina, if he found any new viruses down there.
Oh.
I'm sure he found a lot of fish.
Yeah, he sent me a picture of him pulling in a big fish. I'll have to show you guys that. So next time, we're not going to have TWiV next week, but the following week we'll have Dick back. I hope we can see what he was up to. So let's get right to it. The first story was suggested by Len, who wrote, Vince, I'm forwarding this for two reasons, by the way. It's a mumps outbreak in Israel. First, I think it's an interesting outbreak and should probably be discussed on TWiV. Secondly, is the fact that such a large percentage are reporting 2 shots of MMR but still have become ill. Could we be seeing a shift in the disease that is reducing the effectiveness of the vaccine? If not, what's the cause? Also, remember the large mumps outbreak a couple of years ago in the American Midwest. I recall at the time looking at a CDC report that stated 40% of the infected people had received 2 doses of MMR, and remember wondering at the time whether we are seeing a genetic shift in the virus that reduced vaccine effectiveness. Your thoughts on all of this would be appreciated. Cheers and hi to Dick and Mr. Dove. And, of course, Dr. Dove and Dr. Condit.
Yes.
The mumps outbreak. So there have been a couple of mumps outbreaks, and perhaps we should preface this by saying that mumps is a paramyxovirus like measles and a negative strand enveloped RNA virus. Childhood infection used to be a childhood infection, and then the measles, mumps, rubella vaccine developed by Maurice Hilleman.
Yes.
Got rid of that pretty much, or made it possible to get rid of it. Didn't eradicate the diseases, but significant decrease in their incidence in the U.S.
And do you know where he got the virus to make the mumps vaccine?
Yeah, I do, because I read that biography of him. It's his daughter, right?
Yeah. Yeah, his daughter Gerald Lynn caught the mumps, and he, of course, being Maurice Hilleman, he found out she had the mumps, and he went back to the lab and got some culture plates and some other materials, and came home and cultured the virus from his daughter, and then made an attenuated strain from it.
So that's where the name of the vaccine comes from.
Yeah, and his second daughter was part of the original clinical trial population.
Gerald Lynn vaccine, yep.
Yeah, so talk about putting your money where your mouth is.
Yeah,
Really?
Yeah, you did. It's great. So the incidence in the U.S. went down significantly, but now there have been some outbreaks. There was one, as Len said, in the Midwest in 2005, I believe. I have a paper on that.
2006?
2006. That was sort of concurrent with a big outbreak in the UK. And then there is now an ongoing outbreak in Brooklyn. And now this one that Len reports is from Israel. So,
with evidence that there's a connection with the Brooklyn outbreak.
Yes.
So the Brooklyn
Or-
outbreak seems to have originated from upstate New York, a summer camp. So there was someone with mumps in the summer camp. They seem to have brought it to Brooklyn. And the idea is, I'm not sure if this is proven, that the summer camp was connected to the British outbreak in the UK. So the UK outbreak is a consequence of reduced uptake of the measles, mumps, rubella vaccine.
Right.
Which is a direct result of Andrew Wakefield's publication in the 90s, which suggested that the paper was the vaccine was a cause of autism.
That is the paper that was just retracted.
Exactly. Very, very many years later, in fact.
Yeah, many, many years later.
By the journal.
Right.
Not by Dr. Wakefield.
Right.
Which would, I think, bear more weight if he had retracted it. But
10 of the other authors have bailed out, have retracted, or
yes.
dissociated themselves from it, but not Wakefield himself.
Well, Wakefield is probably still cashing the checks from the plaintiff's attorneys he was working for.
So this is all probably a consequence of him saying, don't take the vaccine. Many parents in the UK didn't immunize their kids. There have been a lot of infections, with mumps in particular, over 50,000, I think, since then, and some deaths of measles. And now it's spread to other areas. So the question...
Really, the global perspective on this is fascinating. I mean, we've got the UK and the US and Israel involved.
Yeah.
It's amazing.
Yeah, so we've got an outbreak in the UK. The biggest one of those looks like it was 2004 to 05. I'm looking at the paper now. And then from there, we think that it went to this camp in upstate New York.
That's what I have read.
And then from upstate New York to Brooklyn.
Yeah.
And from Brooklyn to Israel.
I'm not sure about that. Is that what you're reading here?
The Israeli outbreak, a number of the patients reported contact with yeshiva students from the United States, New York.
Could be.
Who visited Israel during the high holidays.
That's right. You're right.
So it's not necessarily all connected, but it's an awfully suspicious link.
Sure. Now, this is a little peripheral, I suppose, but I wonder if Wakefield knew what he was starting. I have a suspicion he really didn't understand disease epidemiology and what he was about to do.
Well, if you look at the original study, you can tell that he didn't understand disease epidemiology. I mean, it's not an epidemiological study. It's a really crummy paper.
Sure.
But then I think the thing that he really, really didn't understand was how this would end up affecting the public.
Right. Even with the paper retracted, it's not going to make any difference.
No, a lot of people are really entrenched over this.
Yeah.
And if you see the news stories about it, they're on sites that allow people to comment. You see these people who are still irate about it and going on about, oh, this is part of a conspiracy to discredit Wakefield, and it's just...
Who cares about Wakefield? I mean, the issue here are kids who get very ill and die of infectious diseases. And I think the motivation is to protect them. So he had no clue what he was getting himself into. As a gastroenterologist who said, this vaccine looks bad, don't take it.
Right.
Not a good idea. Anyway, this mumps outbreak is interesting, I think, because in all of these outbreaks, many of the people who got infected were immunized with MMR.
With the exception of the one in the UK.
Yeah, so the UK, the biggest one, a lot of those people were not immunized. But in the U.S. outbreak in 2006 and now in this one in Israel, here, 74% of the patients in Israel were appropriately vaccinated for their age. 67% had received 2 doses, and you get the second dose at a certain age. So some of the kids were younger and they hadn't yet gotten their second dose.
My daughter just got her second dose of MMR today.
So why are we getting infection with 2 doses of vaccine? That's the question.
Well, the first most important thing to address, I think, that is the question that was in the email that Len sent, is that does this represent some funny business with respect to the genetics of the virus or something like that? And all of the evidence suggests that that is not the case. Mm-hmm. That the virus that's doing the infection is the same as the virus that's been around all the time. And so it's not like we're seeing the emergence of a new disease or something that's specifically vaccine resistant, or some failure of the some change in the vaccine that's changing things.
Right.
But rather I think what we're doing is learning how effective the current mumps vaccine is, how effective it really is. Because, this is all pretty recent. If you look at the epidemiology, the introduction of the mumps vaccine is on a relative scale pretty recent. Where it was introduced in like 66, and it was sort of slowly introduced in this country, and the second dose recommendation didn't come along until 1989. And so up until 1992, there's a pretty significant background of residual mumps, a lot lower than when it was when there was no vaccine, but still a significant background. So it's only been like, well, I guess only 18 years. But between 92, 93, when it was really down to background, and 2006, we're talking about, 13 years. That's fairly recent. That 2006 blip is a pretty big blip over background. So that does look a little strange. But at any rate, my understanding is that the virus is the same. There's no big difference in the vaccination situation, but what we're doing here is learning about the real epidemiology of mumps and the real effectiveness of the vaccine.
And that 18-year figure that you just mentioned is particularly interesting. In the Israeli outbreak, the median age was just about 15 years. And the only patient who had what I would consider really serious complication was a 19-year-old. So quite possibly a 19-year-old who had not received a second dose.
Well, I think it's worth pointing out before we go further with this that this is a disease that you acquire by respiratory transmission. It's shed in the respiratory tract. You sneeze or cough, you transmit it. The virus enters your tract, sets up an infection, goes into the blood, and then hits other target organs. And the main ones are the salivary glands. And that's the characteristic symptom of mumps, swollen salivary glands. And it can also replicate in other organs. A common one is the testicle. Orchitis, that's called, in 20 to 50% of post-pubertal men. And pancreas, ears, joints, thyroid, heart even. But the point here is that 30 to 40%, a third of the infections are subclinical. You don't see infections. And I think that's an important component here, because it may be that silent spread of this virus helps boost the immunity even after you get the vaccine. So to keep your antibody titers up, one of these papers suggested that if there's no virus circulating, you don't get this natural boost, and eventually your antibody levels go down, even if you've had 2 doses of vaccine. And then if there are enough susceptibles and viruses introduced, boom, you have an epidemic.
Right. So we could even be seeing a little bit of the vaccine being a victim of its own success.
Yes.
Yeah, and I think with the Oklahoma, is that where it was, in Oklahoma in the U.S.?
Midwestern one?
Midwestern outbreak. They said the population density was very low, and that could have been a factor, that there wasn't circulation. Post-vaccine era, very little circulation. So you had an accumulation of people with waning immunity to the vaccine. And the characteristic of all these outbreaks is that they happen in large closed communities of some kind, like a summer camp, or in Israel, a yeshiva, I think.
The Midwest outbreak was college dormitories.
Yes, college dormitories. So that could be it. I mean, we're speculating. We don't know the answer. I don't think anyone does, but those are good possibilities. I think you know someone at CDC who works on this, right, Rich?
Yeah, and I was just thinking about him. I've spoken to him several times over the last few days.
So what's his take
about this? Well, I was just about to mention that one of the things that he talks about is force of infection, which is kind of like my sense is it's an epidemiological equivalent of what we in the laboratory called multiplicity of infection. How much virus you have per person
Mm.
in a given location, so that if you get a lot of people together and the virus gets a grip, you have a center of infection that can spread around amongst those people.
Mm.
But he does. He basically says it's a complex issue that doesn't involve any genetic changes in the strain, and agrees with the notion that we are basically learning about how effective the vaccine is. The mumps vaccine is, although it's quite effective, it is not as effective as, for example, the measles vaccine. What did they say? Something like 85% protection on the first dose and 88 on the second dose. And my contact at CDC says that, in fact, if you look at the real data, there's wide variation in the numbers reported for how effective the vaccine actually is. So it's probably not as effective a vaccine as we had hoped for, and we're beginning to see that. Plus, apparently, immunity wanes. There are also examples of reinfection with wild type people who've actually gotten wild type mumps more than once. So the immunity is not totally straightforward.
Mm. And that of course
So,
can be a virus specific property,
yes,
right? So
and all those things sort of collude to bring about this pattern that we're now starting to see, where we're getting peaks of outbreak under special circumstances over a background which is now very, very low, so that you can actually see this.
So
And
mumps may have a more sophisticated immunological evasion strategy than say measles.
Yeah,
Could be.
make a weak, not a persisting antibody response. In other words, right?
Right.
Yeah, there's one issue that he brought up too, and there's apparently a paper on this. This is somewhat theoretical, but it gives you a way to think about it. Apparently, there's a major glycosylation site. I don't know if it's proven glycosylation site or hypothetical in the wild type virus, where there's an amino acid change in the this is in the hemagglutinin, where there's amino acid change in the vaccine strain, so that site would not be glycosylated. So if that's a major part of the antigenicity of the virus, that's something that would not be present in the vaccine strain, and therefore you can create a story to say that with respect to that particular difference between the vaccine strain and the wild type, the vaccine strain may not be as good. This is the kind of thing that people are going to be looking into.
Sure. By the way, do one of you guys have a cell phone on?
Mine is on. It's silent.
You could just turn it off because we're getting some cell phone interference. I don't know if it's yours, but I'm getting that.
It could be mine. Mine does that occasionally. I will turn it on.
So do we think we could make a different vaccine that would overcome this issue? I mean, if it's an issue with natural infection, it's going to be hard to overcome with a vaccine, right?
Yeah, we don't know what the issue is, so
well, if it's waning immunity, how do you deal with that aside from immunizing people every 10 years?
You could give additional booster doses.
Yeah. Do you think that would be acceptable given the mild nature of the disease?
Well, that's a really interesting point. What is an acceptable background level of disease? And I don't know the answer to that, but it's a really interesting point. Does it have to be zero?
Yeah.
Right.
Or do you just put up with a certain amount of this and say, well, this is good enough, and it's not worthwhile doing anymore? Worthwhile, of course, there's several. Worthwhile, there's lots of units there.
Yeah.
One is economic, one is ethical, etc.
Well, sorry, go ahead.
Oh yeah. I mean, we should also make the point that when we talk about the vaccine failing, there are degrees of failure. I mean, it seems like a lot of the people who are getting mumps who were vaccinated are getting the mild disease. How much immunity do you really need?
Right.
So.
We should point out that in this Israel outbreak and in the U.S., there have been no fatalities.
Right.
No serious consequences, no sterility.
Right.
So
Right.
It's
No permanent damage as
right.
far as I can see in any of these.
But if you can imagine if the outbreaks became more frequent or greater in number, then you might have some issues.
Right.
Right.
Well, not only that, but as we've pointed out, they come up in special epidemiological circumstances. Okay, these conditions of crowding over long periods of time. So it could be that those are situations where public health measures may, in fact, be able to eliminate those or reduce the chances of those problems.
Yeah. I
But
mean,
one of the interesting conversations I've had with my friend at CDC is the notion that, you know, with smallpox, we were lucky. That was a very good vaccine that confers apparently good lifelong immunity with none of these problems. And with most other vaccines where we've tried to approach this issue of eradication and really can get final control on the disease, it's been much more difficult,
Yeah.
each with its own
sure, yeah.
problems.
Well, the original inactivated polio vaccine made by Salk, the initial people who received that in 1955, the 50s, it was found that their immunity waned eventually.
Mm-hmm.
So they became susceptible to infection again. The solution to that was to make a new vaccine which had more potent antigenicity. It had greater and more stable antigen content.
Mm-hmm.
And that solved the issue.
Okay.
So I just wonder in this case if that could be done, if they need more antigen, if they need to reformulate it or purify it in a certain way. I'm not familiar with the mumps vaccine production, but maybe that would help.
It would certainly be something to do, at least at the basic research level, to see if you can generate a more antigenic vaccine.
Yeah.
Mm-hmm.
Because it seems to me that is the issue here, that the immunity is waning, and maybe you could look at that in terms of reformulating the vaccine.
Right.
So as you suggested, a booster might be okay, but if you can avoid a third shot, it would be nice.
Sure.
And simply increase, maybe just simple increase of antigen content would do it. Who knows? So the inactivated polio vaccines are now high. They're called enhanced inactivated polio vaccine.
Right.
Highly purified, more antigen, more stable. So maybe that would do it here. Anyway, measles, mumps, rubella vaccine. That's interesting that in this week we have this story plus the Wakefield retraction.
Yeah, coming together.
So I have seen people, as we said, they're up in arms that this was retracted. You can't silence scientists, but really,
but the science has spoken. I mean, there have been now several enormous epidemiological studies that have shown that Wakefield was full of it.
Right.
Against a poorly structured and now we find out unethical study of 12 kids. There's just
yeah, no
the
question.
issue is settled.
Scientifically, it's very clear, but there are always individuals who don't believe the science for one reason or another and think it's a conspiracy. And there's not much we can do about that, right?
That's right.
Yeah.
That's always going to be
As
there.
the Times says, the arguments were considered by many to be proven earlier, and the ghastly social drama of the demon vaccine took on a life of its own.
Yeah.
So that's the problem, right? It took on a life of its own.
Right, because people have staked out a position on this and they can't admit that they were wrong. Ego is a very powerful force.
And it's not as if the only objections to vaccines that have ever happened are this Wakefield thing. I mean, people have had issues with vaccines since the very beginning, and will continue to do so regardless. They'll find some reason to not like it.
No, absolutely. Right. We are human. We make mistakes. The first polio vaccine, great mistake made with it.
Yes.
I suggest you read the Cutter incident to find out all about that. We actually picked that as a pick a while ago, but the first vaccine for polio had problems, and people died as a consequence. And as long as people make vaccines, there will be issues. Now, maybe if we have robots making them, they would be perfect, but no Cylons making vaccines yet. Okay, I think that's enough for mumps. Thanks for sending that, Len. And before we move on, I'd like to acknowledge the support of This Week in Virology by Data Robotics Corporation, the makers of Drobo. Drobo is a wonderful storage solution for your computer. They're elegant, little appliances, as we called them last time. They're beautiful black boxes into which you can put one or more drives, hard drives, to store your data on. And they have multiple hard drives, and the data storage is redundant. So if you ever lose a drive to failure, which will happen, you simply pop it out and put a new one in. And they come in all different sizes. Go over to drobo.com/TWiV. They've made a special landing page for TWiV subscribers to check it out. I've been using them for years. They're terrific, reliable. I have a couple of them at home, and I've just got one here at the lab where I'm putting all the old data, all the old theses from my former people, including Alan Dove,
Oh dear.
is going on the Drobo, all the old data, all our protocols, nucleotide sequences, and I'm putting it on the network so the people in the lab can all access it. It's really exciting. And Dick Despommiers just got one. We're going to put all his photographs on it. Now, Drobo has been very nice to us, not only sponsoring TWiV, but they're providing 3 Drobo S models. This is the 5-drive model to give away to listeners of TWiV. And we're going to give one a month away for the next 3 months. You can go over to twiv.tv/drobo-contest to read all the rules. Basically, what you have to do is subscribe to TWiV in iTunes and leave a review there, and then find my original tweet about the contest on Twitter and retweet it. All the rules are there. These are great models. The Drobo S has a very fast interface, so you can actually do work while your data are stored on the Drobo. So we have 3 to give away. You can just subscribe to TWiV to enter the contest. No purchase necessary. So you might win a Drobo, and you'll learn about viruses in the meantime by listening to TWiV.
Alan, how does it feel to know that you're reliably backed up?
Well, now that's a personal issue.
Yeah, right. He's reliably backed up. Yeah, well, my former students' theses are on the Drobo. They'll be there forever.
So you have a big pile of theses.
Big pile of theses. Yes, keep the T there. So Drobo, we're really grateful to Drobo for supporting TWiV and also for making this contest, and it'll help build awareness for Drobo in a new area, people who are interested in viruses, and also help more people to learn about viruses by listening to TWiV. So we thank Drobo for their support. Our next story just came out in PLOS Pathogens, one of our favorite journals, because it's open access. And anyone can go read this article right now. It's called Immunization with 1918 or 1976 Swine-like Viruses Protects Against 2009 H1N1. So this is a paper which I've been waiting to see for a while because one of the authors, Peter Palazzi, had mentioned this result last year. In fact, last summer, he said they found this. And here's the background. Everyone knows that we have a pandemic influenza strain circulating. Is that right, guys? You know that?
Yeah.
I'm aware of that, yeah.
I think I had heard 1 or 2 things about that.
This is quieted down quite a bit. In fact, we haven't talked about it on TWiV for 2 weeks, I think. And the numbers have gone down. I'm actually surprised that we don't have more activity at this time of the year.
Yeah, so
Let's check
when
it out.
officially is flu season?
Now.
Right now?
Absolutely, right now. So if you look at last year, it begins like January. Let's look at a graph. Go to cdc.gov, and we're going to go look at flu activity, and we have flu view. So at the moment, it's last week, 3,500 specimens were tested. 164 were positive for influenza, most of which were the new H1N1 strain. No H3s and no H1, seasonal H1s. So that's very low activity.
Right.
And in previous years, it would have been going up already. But we had a peak already at week 42. That would be November, roughly. And that's unusual because this was a pandemic strain. And now it's pretty flat. So if this were seasonal flu, it would be going up already. So I don't know why that is. I don't know why we don't. Maybe a combination of extensive spread and immunization has immunized a good fraction of the population, so you can't get spread anymore. But anyway, the interesting observation about this novel H1N1 was that it seemed that people over 65, which are usually really susceptible to flu and can have complications, are pretty much spared. There's a lower level of infection in the older population. And young people seem to be more susceptible to the 2009 H1N1. So the authors of this paper wondered whether that's because old people were previously infected with viruses that provide some protection. So what they did is they took mice, and if you infect mice with the 2009 H1N1 virus, they die. It's quite a lethal infection in mice. And if you first give them a low dose of the strain and let them develop antibodies and then challenge them, they're protected. So in other words, we establish a lethal challenge model in mice for this strain. Okay. Then they took a variety of viruses from 1918 to the present, actually, all H1N1 viruses, and immunized mice with each of those and then challenged them. So they took a 1918, actually virus-like particles from 1918, so they wouldn't have to do this in BSL-4 facilities. I don't know what you have to work with the 1918 strain in, but high containment. So they immunized mice with virus-like particles from 1918, then a few H1N1 viruses between 1918 and 1943, and then some contemporary seasonal H1N1 viruses from more recent years. What they find is that the H1 viruses from 1918 to 1943 confer protection against infection with the 2009 strain. But contemporary H1N1 seasonal strains like 1977, they don't confer any protection. Okay, now it's worth pointing out that in 1918, the H1N1 strain went into people and pigs at the same time. So that virus evolved in pigs along with it evolving separately in people. So then they took a few swine isolates over the years, from the 30s and the 70s. They even took the 1976 swine virus that jumped into a number of people at Fort Dix, New Jersey. They also immunized their mice with those, and they get complete protection with those strains as well. Everybody with me so far?
Yeah.
So this explains why old geezers like us, well, we're not over 65,
Yeah. Well,
are protected, because if you lived up until 43, or if you got immunized with the 1976 Gerald Ford swine flu vaccine, you're protected against infection with the 2009 strain. And if you're younger, you weren't infected with those viruses and you don't have protection. So they say young people should definitely get vaccinated with the swine origin 2009 virus. The other thing that's cool about this is that, so the virus in people evolved from 1918 to 1943, and then it kept going to 1957. But from 43 to 57, it obviously was different enough so it no longer confers protection against the 2009 strain.
And so was H1N1, that wasn't the predominant strain all during that time,
It
was
was, yeah.
It was.
From 18 to 57, yeah, very long run. Long run on Broadway.
And then there was a new pandemic that was what, H3 or something?
H2N2 came in 1957. Then H3N2 in 1968. Then
and those two effectively, as we're seeing now, displaced the pre-existing?
They did. And then in 1977, the H1N1s came back, probably from a freezer from the 1950s. Mm-hmm. And they've been circulating ever since until last year.
Co-circulating with...
Yeah, they co-circulated with H3N2. Those viruses don't confer any protection against the 2009 H1N1. So only the H1N1s up until 1943, because apparently beyond that they evolved to be different enough. But in pigs, you can go as recently, let's see, what was the most recent pig virus they used? I think it was 1976. Swine. So they used the 76 swine-like strain. And those viruses, the idea is that they evolve much slower in pigs than in people.
Right. That's the interesting thing about all this for me. Somehow the evolution in pigs is different than in people. Why is that?
I don't know. Nobody really knows. I don't know. We'd all be guessing. Nobody knows. But basically they're saying in this paper that pigs are a great source of viruses because they stay pretty much the same. And as we get older, we lose immunity to, similar viruses that we've had. And then, boom, they come back out of pigs again. So in, 60 years or 70 years, the virus that's in pigs today could come out and cause another pandemic, or part of that virus anyway.
I mean, just to throw out a couple of guesses about why the pig viruses might be more stable, it's at least in domestic hogs, they're going to be a few inbred populations. They're going to have a high turnover rate. They're being slaughtered on a regular basis and replaced.
Hmm.
I mean, I could expect that might be an environment where the virus would not need to change much in order to find new hosts.
And in particular, the inbred characteristics. That's an interesting thought.
Right.
So there's something that's bothered me for years about all of this, and it may be a no, it's probably a technical thing. So I understand that an H1 strain, for example, can undergo what we call antigenic drift, which is basically what we're talking about here in the human population between 1918 and
43.
43.
Right.
There was drift in these, so that the earlier viruses don't necessarily completely, well, actually, between 1918 and 1957. Okay, so the 1957 virus doesn't cross-react with the 1918 virus, right? Because it's drifted so
Yeah,
far.
you wouldn't get protection, right?
Right. But we still call it, what are the criteria for calling that H1?
Well, it used to be antigenic. You would make antibodies and see if they cross-reacted.
Right, but now we're talking about drift that's so exaggerated so that there's no cross-reactivity.
Well, in
Okay?
fact,
And yet it's
yeah.
still
Yeah, so you can have two H1s that don't cross-react at all.
right. So how are they called H1s?
Well, now
Is that
it's done by sequence. You compare the sequences and you say, it's an H1 sequence.
So we have sort of arbitrary boundaries. If it differs more than this amount, then it becomes a different serotype.
Yeah. I mean, so you can see that the sequence can differ significantly so that it doesn't give you cross-reactive antibodies, but it's still an H1.
okay? So it used to be serological, but now we're talking more like
Yeah.
sequence.
It
Okay.
is sequence. But, you can't predict the serology from the sequence. That's actually an interesting problem. There's a fellow in England whose name is escaping me, but he developed a technique called antigenic cartography. And it's an attempt to predict the antigenic property of a protein by the sequence. Because right now, if you look at an H1 sequence, you can't really predict whether it's going to react with a given antibody or not,
Mm-hmm.
depending on what changes have happened. The only way you know that is by doing the experiment. So you can use the sequence and say it's an H1, but if you want to know if it's cross-reactive with another H1, you have to do the serology still.
Right. Well, I guess predicting the antigenicity is like trying to predict the three-dimensional structure from the sequence.
Exactly. My
yes.
thoughts exactly.
Exactly.
Which is one of the toughest problems in computational biology.
Right. And we're working on it, right?
Yeah.
And someday we will, but not now.
Not quite.
It's worth pointing out the 1943 H1N1 did not protect as well as earlier H1s for these mice that were immunized. So there is drift there already. Right. So it's not like it stayed the same in people from 1918 to 1944. It was drifting, and there was less and less protection as they moved through the years. But apparently in pigs, the H1 that's in pigs today is very similar, is similar enough to protect.
So it's interesting because, the sort of the dogma that I preach, or have preached at any rate, is that birds are the primary and most significant reservoir of virus, in particular when it comes to pandemics. And while they're certainly maybe even the most significant reservoir, and certainly most of the influenza viruses are in birds, this sheds quite a different light on pigs. Pigs are a very significant reservoir for what finally winds up in humans.
Yeah, and we sort of ignored the pigs. We said they were a mixing vessel, but now it could be that that's where the hemagglutinins originate. Sure, or could originate.
And do I remember that at the end of this paper, they actually have some comments on the impact of these studies on, or what it means for raising pigs?
What they say is the surveillance and containment of swine influenza viruses is desirable for the prevention of future pandemic episodes.
There you go.
So we should look more carefully at pigs.
Right.
Actually, we have an email later, which we'll get to, from someone who has comments about pig farms, which is timely for this. Anyway, PLOS pathogens, check it out. It's a neat paper. It's from the Palese, Bassler, Garcia, Sastre groups at Mount Sinai School of Medicine. where I got my PhD many years ago. And our last story, this is just great. Chikungunya virus. A virus-like particle vaccine for epidemic chikungunya virus protects non-human primates against infection. Now, when I was a PhD student at Mount Sinai in the 1970s, I heard about chikungunya virus. Chikungunya, by the way, means it's derived from an East African dialect describing the contorted posture of patients with the severe joint pain characteristic of the disease. I love the name. I thought it was cool.
Yeah.
Chikungunya.
Right.
So not chicken.
Yeah,
Chikungunya.
right. Chikungunya. I loved it, but at the time in the 1970s, it was a pretty exotic disease. Now, this virus was identified in 1953, which happens to be a wonderful year. And joint pains, severe joint pains, rarely fatal, but, it infected a lot of people in Asia and Africa. But in the intervening years, since I was a student, the virus has spread. And the reason is fascinating. So originally, this virus was transmitted by Aedes aegypti, pretty much a human-specific mosquito, if I'm not mistaken. Correct me if I'm wrong.
Yeah, this is where we need Dick.
Yeah, Dick's not here. No, it's not true. But in 2004, there was an outbreak started in Kenya and spread to islands in the Indian Ocean. And in fact, Réunion hadn't been, and in India, eventually reached India, where it hadn't been found for 32 years. So Réunion, one of the Indian Ocean islands, 40% of the population got infected with this virus. And it spread to Europe from a traveler from India. And so it's spread quite globally. It's not in the U.S. yet. It's been brought to the U.S. by travelers, but I don't believe we've had transmission yet. So why did it change? This is fascinating. Apparently, the virus made a single amino acid change in one of the glycoproteins in the virus particle. And this now allowed the virus to efficiently grow in a different mosquito, Aedes albopictus, which it so happens to be the predominant mosquito in this Réunion island in the Indian Ocean. So previously, the virus could grow in this mosquito, but not very well. So it didn't transmit efficiently because it bites a lot of different kinds of animals. So the idea was it doesn't make a lot of virus and it gets diluted as it bites different species. But this amino acid change makes it grow so much better that now it effectively transmits among people. And I think that's great. One amino acid change of vector, now the virus is spreading all over.
Right.
And this mosquito Aedes albopictus is spreading all over the world because of they use tire trade, right? The tires are put on big ships and they have some water in them, and the mosquitoes are breeding in the water. And in fact, this mosquito used to not be in the New World. It came here in 1985. It was first found in Houston, where it probably came from Asia in a boat carrying scrap tires. So it's almost all over the U.S. at the moment, and it's also a vector for other bad viruses. So I guess eventually it'll be here in the U.S. So with that background, this paper reports a experimental vaccine, which is pretty neat because it's not an infectious vaccine. It's a virus-like particle vaccine. So chikungunya viruses are alpha viruses. They are enveloped viruses with glycoproteins in the envelope. And within the envelope is a icosahedral nucleocapsid composed of one protein called C. And within that is the plus-strand RNA genome. In this paper, all they do is take the genes encoding the capsid protein and two glycoproteins. And they express those in cells, and the cells make virus-like particles. So these are empty particles with no genome in them. And then they immunize animals with them. Eventually, they immunize monkeys. They make antibodies that protect them against infection. And then they can take the antibodies out and put them in mice. They take the monkey antibodies, put them in mice, and they protect them as well. So I think it's pretty neat. We've talked about virus-like particle vaccines before, and here's one just being developed because this virus is spreading, and this is probably going to be needed soon.
Yeah, well, it's important because the same strategy theoretically can be applied to a number of related alpha viruses that are a significant problem as well. The encephalitis viruses. What is it? Venezuelan equine encephalitis
Yeah.
virus and several other
Yeah,
similar viruses that I would assume the same strategy could apply.
absolutely. They're all similar structurally. So Western, Eastern, and Venezuelan equine encephalitis viruses. I think
And
we've,
from a manufacturing standpoint, this is a great way to make a vaccine.
yeah, there's no danger, right? You don't have to
Right.
worry about the
There's
infection.
there's no danger of infection with it, and you can also, if you can stably transfect cell lines with it, industry is well geared up to grow large quantities of cell products.
Right.
So you wouldn't have to deal with uh, some of the things that come up with flu vaccines, for example, with egg supply and all that. It would just be big incubators of cell culture and
Yep.
purify your vaccine from them.
Yeah.
Coincidentally, just the other day, I heard a seminar from Scott Weaver from Galveston,
Hmm.
who was working on live vaccines for these same viruses, the encephalitis
Live.
viruses and chikungunya, and the problems. I mean, obviously, the motivation for doing that is that. The dogma, at least, is that live viruses give better immunity than the subunit vaccines or the killed viruses. Um, and but the problems associated with creating the live vaccines are huge. First of all, as Alan's already mentioned, there's the biohazard factor. A lot of
Right.
these viruses are high containment viruses, so all the research and development has to be done under high containment. And you have to worry about reversion. And you have to worry about whether or not the thing that you make is going to, even if it's relatively safe, is going to replicate in mosquitoes. And so there's very significant problems associated with making the live vaccine, as desirable as that might be.
Sure.
And none of those problems are present with this.
They actually summarize the efforts, previous efforts here. They say there has been limited success in developing a safe chick vaccine. A live vaccine candidate caused transient arthralgia in volunteers, and formalin-killed vaccines have not yet proven safe and effective. So yeah, this offers some advantages. The thing I've noticed about these virus-like particles that they can be even more. They're more immunogenic than subunits, obviously, because they're intact virions.
Mm-hmm.
So they may be good. They may give durable immunity. We do have two existing virus-like particle vaccines in use already. Right, that would be the hepatitis B virus vaccine and the human papillomavirus vaccines.
Right.
Right,
Right.
they're virus-like particles made in various cell systems. They work pretty well, although the HPV is relatively new, so we don't know how durable immunity is yet.
Right.
And we have talked about making virus-like particle vaccines against flu before here on
Right.
TWiV, particularly in plants, if you remember. There's a company in
Right.
Canada who's making these VLPs in plants, and they are very immunogenic for flu, at least, and very cheap, 20 cents a dose.
Right.
And if you remember, a square meter of plants will give 20,000 doses of flu vaccine. That's amazing. So if you can
There's a lot of cool stuff to be done here. This is great.
yeah, if
Yeah.
they could do this in plants, wow.
I wonder if you could, well, I guess if you can make the flu virus-like particles in plants, I don't see any reason why you couldn't do this one.
No,
Right.
nothing obvious. No, right. It's a few viral proteins induce budding of a particle, and that's all you need.
Yeah, and I
So-
think there has been some progress lately on plant transfection to the point that that's gotten easier as well. So somebody really ought to do this experiment.
Yeah, I'm sure that they will explore it. It's clear. It's obvious. Yeah, it's very cool. Anything else on that, gentlemen?
No, I don't think so.
Chikungunya.
Chikungunya. We just want to say chikungunya a couple more times.
An exotic virus on the move, folks. It's on the move.
Getting less and less exotic.
Yep. It really isn't very anymore, is it?
Hmm.
Shall we move on to email, gentlemen?
Sure.
Sure.
And save our fourth story for next time, which sounds great. Okay, Attila writes, after listening to almost all TWiVs, a recurrent question is about pathogen virulence evolution. So I would like to contribute with some information. Paul Ewald, an evolutionary biologist who proposes that many human diseases may be caused by germs, not genes, otherwise they would be ruled out by natural selection, has addressed the question of pathogens' virulence. He argues that pathogens may evolve to be more or less virulent according to their transmission routes. So pathogens that depend on host mobility, like influenza, should be selected to be less virulent, allowing its host to move around. But pathogens that don't depend on host mobility, like vector-borne diseases or long environment survival pathogens, would benefit from a higher virulence and increased viral loads that favor mosquito transmission.
I think we've talked about Ewald's work on this.
Have we?
Yeah.
Yeah,
Or
at times.
it's certainly something that I'm familiar with. It's a very interesting idea with a fair amount of fair number of examples, and I guess Attila gets to one of those.
Well, so another case he mentions is cholera in South America, places that have unprotected water supply that increases diarrhea transmission have high virulence circulating strains that cause severe symptoms, while places with protected water supplies have low virulence strains that persist longer in the host and increase contact transmission. So selection favors the most adapted pathogen, whether its transmission is favored by higher or lower virulence. So he gives us a couple of references by Ewald. So actually, this email came in a while ago, so I picked up one of Ewald's books, and I'm reading it at the moment. So we've always argued about this on TWiV, or discussed it, and I've always said, well, there's no point in a pathogen becoming more virulent unless it helps transmission. So I guess this is consistent with
That's
that.
consistent with that.
Yeah.
One of the stunts I like to do in lecturing medical students is to point out to them by taking a little on-the-spot survey, point out to them that they're all infected, or probably all of them are seropositive for 3 different herpes viruses. HSV type 1, the virus that causes cold sores, EBV, that causes infectious mononucleosis, and VZV, that causes chickenpox. And I have them raise their hands as to whether before I reveal that they've all been infected, raise their hands as to who's had either cold sores or mononucleosis and chickenpox. And of course, out of a class of 100, there's 10 or so that have had cold sores, and another 10 or so that have had mononucleosis. And then everybody raises their hand when it comes to chickenpox. So these are viruses that are in many ways pretty similar. But in the case of the first two, they very rarely, only 10-15% of the time cause clinical symptoms.
Right.
But in chickenpox, it's almost all the time. And I've only recently come to think that, maybe that's because the lesions in chickenpox are important for the transmission. Okay.
Mm-hmm.
So it's a benefit to the virus to give a full-blown, obvious clinical symptom like a rash in order to promote the transmission, whereas the other viruses can do it in saliva without giving a full-blown disease. So
Right.
that's consistent with this notion that transmission is an important factor in determining what the pathogenesis of the virus is going to look like.
Nice. So he gives us also a PLOS pathogens review, crossing the line, selection and evolution of virulence traits. So we'll post those because those are very interesting. I'm reading, and as I said, I'm reading Ewald's book, and we'll come back with more discussion about this issue, because a lot of people feel that viruses evolve towards higher virulence, and that's the end of the story. And I'm not sure. I know that's not correct.
No, they evolve in whichever direction favors transmissibility.
Transmission, exactly. That's
Right.
the bottom line. It's
Right.
transmission.
And for some viruses, that will be less virulence, and for others, that will be more virulent.
Right.
Quite frequently, it's less virulent. There's a lot
Yes.
of viruses out there that we really are mostly not aware of.
Right.
Right.
Because
Raise your hand if you're CMV positive.
Right.
Yeah.
I'm not going to be able to do that stunt with the medical students much more because of the chickenpox vaccine.
Yes,
Right.
that's right. Yeah, you're right. Hey, I used to do it with measles, and I said, "You're all probably infected with measles," and I realized that everybody had been immunized after a certain point. I said, "Oops." He notes here that flu should be selected to be less virulent, and I think that's interesting because in the last year, many people were saying, "Oh, it's going to become more virulent and become a problem," and there was no reason to assume that it would do that
No.
at all.
No, in fact, quite
Right.
the opposite. If you get a mild cold and you're likely to go to work, then that's that many more hosts.
Exactly. Exactly. Whatever transmits, you're right. So thanks, Attila, for contributing. We appreciate it. The next email is from Mike. He writes, I am a lab rat with the company that makes flu mist. I've been with the company for 5 years now and try to find all the resources I can to improve my career. I was searching around for a podcast about viruses for a while now, and I'm very happy that I found yours a couple of months ago. I've caught up on all the previous episodes, and now one a week just isn't enough. It's a great podcast, and I hope you keep it up along with TWiP. Any takers on TWiB yet?
What's TWiB?
I presume it's This Week in Bacteria,
I
guess.
Okay.
We'll try and keep it up. As long as people listen, we'll keep it up. If no one listens, then we won't keep it up.
Right.
We'll just talk to each other whenever we see each other.
Right.
I have a quick question that's been rattling around my brain for a couple of weeks with this H1N1 pandemic. It's well known that it affects those under 25, hypothetically because those over 25 have been exposed to enough virus strains to build up a better immunity. If this is true, wouldn't it make sense to get vaccinated every year? This way, even if the person doesn't get flu that year, that person has built up immunity to that and future strains in vaccines, which would make a global pandemic less likely. When the next novel strain hits, more people will be better equipped and less likely to pass the virus.
Yes.
It depends what kind of vaccine you get every year. So
Right.
what he's talking about is cross-protective immunity, right?
Right.
Right.
And if you get a killed vaccine, that doesn't induce cross-protective immunity because the epitopes are gone. Those are internal proteins that are destroyed in the production. So flu mist, yes, your company's product, it's great. It probably induces cross-protective immunity because it's infectious. So you should get that every year, and that might help long-term immunity. Now, interestingly, this year, the reason why older people are protected is not only because of this, but as we just talked about in that paper, it's because the virus is related to ones that circulated years ago.
But also, if you even with the killed vaccine, if another strain comes around that is related to a previous vaccine strain,
Right.
you could get some cross immunity to it.
Sure. Right. So I don't know if he's distinguishing between those in his question, but
Right.
yes, so there are multiple mechanisms. It could be
So-
recirculating strains or cross-protective immunity.
I'd never thought about this thing about the difference between the killed and the live vaccine relative to cross-protective immunity. That's interesting. So it's the nucleoprotein or something?
Yeah, nucleoprotein M proteins. They think these are T cell epitopes that give you broad protection.
Because they're more highly conserved than
Yeah.
They don't drift like the hemagglutinin and the neuraminidase. That's interesting.
Right. And the
Right.
kill vaccines or the inactivated vaccines don't have any of that pretty
Right.
much because it's destroyed. So, in fact, there was, if you remember, Alan, a paper out of Canada a few months ago saying it was a bad idea
Yes.
to give kids killed vaccine every year because it prevents them from getting this cross-protective immunity. And
Right.
so they're saying, to protect them against the future pandemic, you shouldn't immunize them every year. Which
The problem being that then they're not protected against the seasonal strains, which can
Right.
also kill them.
Right.
And this is a theory. We actually don't know if this is true or
Right.
not. It has some support in animal studies, which we actually talked about some time ago. But I think you don't, hedge your bet for the future and ignore what's going to happen at the moment, right?
Right.
So anyway, Mike, thanks for writing. He writes, thanks for reading and keep up the excellent work. I appreciate you taking the time and effort to educate the masses. Well, we try. We need more people to listen. We want to educate more people. Tell your friends about TWiV. Cheryl writes, hi, I was just listening to the new TWiV where you were talking about the paper speculating about the origin of swine origin H1N1. I wanted to point out a problem with the notion of flu not being able to get out of these pig farms and traveling around. I'm neither a virologist or a scientist, just a very interested layperson. However, I grew up around farms.
This is great. I love this perspective.
This is what we need because we don't know this stuff. Right?
Yeah, this is great.
Sitting in our ivory towers. This is referring to a paper which we talked about last year, which suggested that the H1N1 came out of a lab. And the idea was that it couldn't possibly have come from a pig farm because there's no way all these strains could have gotten together. This was a paper out of Australia, Adrian Gibbs's paper.
Right.
So she continues, 1. While in big commercial pig farms, the pigs themselves are fairly isolated, their waste is not. This includes uneaten food and manure mixed together, and birds are very attracted to this waste as a source of food. If there is any virus in that waste, it could be passed to the birds. 2. Large commercial pig farms only comprise part of the pig population. Large numbers of pigs, both in the U.S. and even more in countries with more agrarian populations, are kept on small family farms in the open and in close proximity to domestic and sometimes wild birds. These pigs also travel more as breeders send piglets out to farmers who want to raise only 1 or 2 pigs, or bring their boars to sows at other farms for breeding purposes.
I know a joke about that, but I won't repeat it.
It's not a family joke?
No.
Oh, okay. This is a potential population for these viruses to hide, since they are rarely tested for disease. 3. It's possible if the virus arose in Mexican pigs, their farming practices, even on large farms, may not isolate the pigs as strictly as in the U.S. These are just a few of my thoughts. I don't think it's anywhere near as difficult for this virus to have arisen naturally as the author of that paper from my experiences with farming. Cheryl. Yeah, it's great.
That's a great perspective.
Yeah, that's what I think we sort of hinted at a couple of those arguments, but it's great to hear it from somebody who has seen this at firsthand.
Right. So I'm really glad we're reaching many different people and we can get their input on this stuff because this is great. So thanks very much. Peter writes, "Hi guys, I'm not a virologist, but an IT engineer. I do, however, have an interest in viri from a robotic perspective. Anyway, I was watching a show called Viruses Sell Pirates, and at one point I heard a voice that made me look up. I know that voice. I had to rewind to get to the caption. Dixon de Pommier. Was that Dick from TWiV? A quick look at iTunes confirmed it. So Dick, more famous by the minute. Grin. I don't know this episode. I have to look it up.
Yeah.
I am doing some work on computer viruses and contrasting that field with real viruses. There's some really interesting stuff coming out of this. What nature is doing with sheer numbers and genetic error, programmers are doing with their own trial and error. The attacks, vectors, defense techniques, and even epidemiology of the two make you wonder if there are only a few really good ways for viral or infectious behavior to occur, albeit man-made or natural. Anyway, keep up the good work. Thanks for all your efforts. Long-term listener, Peter." From Australia.
And actually, I've kind of wondered, Peter, maybe you can do this experiment in some closed network. If one could set up a computer virus in some language that can alter its data structures like Lisp, and actually just let it evolve and see if you can mimic the behavior of real viruses and their transmission patterns with a program. But you'd want to do that in a network that's not connected to the internet, I think. Please.
It's, I'm sure people do that. I'm sure. I mean,
because there are computer languages that can. Lisp is the most famous one that can actually the program can rewrite itself, which would give you the basis for evolution.
Sure,
would seem.
Sounds like a great idea. It sounds like a great model for studying viral evolution. Right?
I'm sure somebody must be doing that, but
maybe someone listening can tell us about it.
Yeah. I'd like to see Ewald's principles applied to this whole same thing.
Absolutely, yes. It would be interesting. Very interesting. I'm not thinking well about it right now, but
I'll get my son to listen to this. He's heavy into this.
Just do be sure that you have him unplug the cable modem first.
Ken writes, "If you ever think of starting a line of merchandise with bumper stickers, you could do a lot worse than something Dick said today. Quote, safecrackers. They're all safecrackers." I guess he was talking about viruses, right?
Yes.
It's a great line. I like that. And finally, let's wrap up with some more emails from the virology class out in Indiana. Heather writes, Dear
No,
Vince,
in Pennsylvania.
Indiana University of Pennsylvania.
Yeah.
Is that right?
Yeah.
Yes.
Sorry. IUP.
It's a
Heather.
little like Miami University of Ohio. Sorry, continue.
Right.
Heather writes,
I was wondering how the retroviral assembly process controls the number of RNA strands that are incorporated into the capsid. TWiV is a great site. I really enjoy your conversations with Dick. Thanks. Heather, pleasantly perturbing paramecium at IUP. PPP. Well, there are 2 RNAs in every retrovirus particle, Heather, and the way they get there is that in order to be packaged, the 2 of them have to form a structure, a secondary, tertiary RNA structure that interacts with the capsid protein. And that structure only forms when the 2 RNAs come together, right?
2 and only 2.
Only 2. It's called a, what's the name of the damn thing? Anybody know the name?
Mm-mm. Sorry.
Dimer linkage sequence. There you go. It's a little structure at the end of one of the RNA. So each RNA by itself makes a structure, but it very weakly interacts with capsid. But when the 2 come together, the 2 RNAs interact, and they're called kissing interactions, stem loop type things. They form a structure that's recognized, and that's how it's packaged. Good question. Emily writes, how is it that retroviruses have so many tactics for survival? What has caused them to evolve in the many ways they have?
the immune system, for starters.
Immune system, looking for ways to be transmitted effectively.
Yeah, there's an ongoing war between the virus and the cell. Each one is trying to win. And in the process, they try just about everything and wind up with lots of different tactics.
Yep.
Not just retroviruses, viruses in general.
I think
Yes.
that's it. Probably all the viruses are pretty much the same, right? They all have a
Mhm.
lot of tactics. They look for new hosts. They make a lot of mutations so they can evolve quickly. So I wouldn't make it specific for retros, even though they're very interesting. They get a lot of attention because they do cool things, but other viruses too.
And
Right.
all life, if you want to generalize
But viruses are not life.
it.
Oh, here we go.
Well, Alan thinks they are life, right?
I think depending on your definition, they could be considered alive.
Okay.
Have we looked back at that survey recently? Are people still plugging into that?
I don't know.
Last I looked, there was like 390 responses.
Oh,
or
we have
something.
over 500 now.
Oh, okay. What's the score?
Let's take a look. We'll go look right now since we can do that and remove it in post. Okay, waiting for the page to load. Okay, we have 510 responses. Yes, 28%. No, 33%. No is pulling ahead, but something in between is 32%. So the yeses are losing, but I don't know has stayed the same, 7%.
All right.
I don't know is on second, right?
Yes.
Yes.
I see yesterday's pitching.
Yesterday. What a great routine. All right. Kelly writes, on podcast number 66, it was stated that there was homology between hepatitis B and HIV due to a common ancestor. What was the common ancestor and how was it determined? I think what we were talking about was there is homology. Both viruses have reverse transcriptases.
Right.
And they have
Right.
some homology, and which suggests that they had a common ancestor. What it was, who knows, right?
Right.
Yeah.
I mean, we don't have any fossils. That's the problem with viruses. All we can do is look at the sequence and make estimates of evolution and say, yeah, it looks like they had a common ancestor. So there are sequence ways to do that, which involve calculating rates of change and so forth. And so you can say, they're related and they had a common ancestor, but we don't know what it is. The interesting thing is that hepatitis B is a DNA virus and HIV is an RNA virus, yet they both have reverse transcriptases. So very different viruses, they have this common protein. So maybe there was an ancestor to both viruses and they diverged at one point. I don't know.
If you look at the life cycle of the two, it's essentially the same. It's just that HIV and other retroviruses package the RNA in the cycle, and hepatitis packages the DNA. Depending on your, Don Gannon was called hepatitis B, a retrovirus with a bad sense of timing.
That's fabulous.
That's excellent.
Don is so funny.
He is.
He could be a real gas on this podcast.
He would be.
Yeah.
Shari writes, hello, upon listening to TWiV on reverse transcription, I wondered, since it had been found that there is a virus in chimps, SIV, that is similar to HIV and progresses to cause AIDS-like illnesses, does this virus also use reverse transcriptase when infecting the chimps? And why did the chimps only experience similar symptoms if chimp and human genomes are so similar? Shouldn't the symptoms be almost identical?
Well, chimps and humans, they have a lot of genes in common. They're quite similar, but they're still quite different. And the viruses have evolved in those individual niches. It could go anywhere. We're talking about.
It depends on
a lot
which
of
humans and which chimps you're talking to.
I was waiting for you to do that. Some humans look like chimps,
or more to the point, act like them.
Act like them. Yeah, the little bit of difference is enough to make you respond very differently. But in fact, the diseases are probably very similar, right, Alan? The AIDS-like disease in these chimps. But it's not easy to study because we can't capture them and study them, right?
Right.
So we only look at them, we take their urine and say there's virus there, and then we see that they die or disappear. So we say it's AIDS-like because we simply can't study it. But it may, in fact, be more similar than we think.
Right. Well, a couple of the autopsies they did on chimps that apparently died of this disease made it look quite similar to the human disease.
But so I think if you could study it in real time, you could have a better idea, right?
Right. And the manifestations are going to be different because most humans are not swinging through the trees and doing the same sorts of things and getting the same exposures that chimpanzees are.
Sure. But they both use reverse transcriptase. Yes, they're both retroviruses. Yes. That's for sure. All right, that'll do it for email. Let's move on to some wonderful picks of the week, and let us start with Rich.
Okay, so I don't know if this is stretching the definition of science or not, but I don't care.
I've gone pretty far out there with my picks before. So.
It's okay. You can do whatever you want.
All right. So I'm going to plug a local hero, a gentleman named John Moran, who is a nature photographer who got spent a lot of his career, I don't know if got his start is right, but spent a lot of his career with the local newspaper, the Gainesville Sun, and had a lot of nature photographs in that. And several years ago, set out on his own and is now an independent freelance nature photographer. And I've included a website here to his stuff. He takes absolutely the most beautiful and most fascinating pictures of Florida. He's almost exclusively Florida. They aren't really well represented on this site. You get an idea. He also has a book that has a compendium of his photos in it that's not too expensive, and you can buy the individual prints. I typically give these prints to students when they graduate. I think that something like this, nature photography, gives people an awareness of their environment, their surroundings, that's really important, and anything that sort of increases our sensitivity to the relationship that we have with the planet, I think is important to promote, and that's the scientific spirit in which I promote John Moran's stuff. This one that shows up on the very front, it's called Fireflies on the Itchitucknee. The Itchitucknee is a spring close by, and he's got a picture that he must have taken over open the shutter for a long period of time to get the light right, and you can see these green flares going across. You can see where the fireflies were flying. I didn't know.
Cool.
That's like little dash lines and stuff. At any rate, they're beautiful pictures.
Yeah, it's science. It's nature. No problem.
Yeah, this is, you're saying it's not a great representation of his work, but I was just flipping through these on the site. They're spectacular.
Yeah, they're beautiful pictures.
I like this one of the alligator with this baby on it.
Yes. Yeah, that's great.
That's a famous one.
Is it? Yeah. So would you say that Florida is a beautiful state?
Yes, absolutely. There are there are parts of it that's kind of scrub and stuff, but there are lots of it that are actually quite beautiful. The coastline is beautiful. It's its own kind of beauty. It's different than the Rocky Mountains or the Appalachians or the Pacific Northwest. It's a very unique sort of environment, but it has its own kind of beauty that's very well represented in Moran's photos.
Nice. I see you can download that cover as a pretty big image for your desktop. That's
Yes.
nice. Where is he based in Florida? Do you know?
Actually, I'm not sure. It may even be still in Gainesville. I've actually met him. He sets up a booth at our local Spring Arts Festival. There's a show at a town close by. I know he's lived in Gainesville for a long time. I don't know if he still lives here or not.
Now, we ought to email him and tell him we picked him.
Good idea.
He should listen to TWiV because he'd probably like it.
We'll do that.
That's cool. That's very beautiful. I love it. That's why, I mean, every now and then we can all pick local stuff like this. I'd love to hear about it because this is something I would never discover independently. Yeah. Very nice. Alan?
I have picked a chemistry pick. It's the periodic table of videos. Now this is
front page is a gas.
Yes, it's great, and I think that guy on the right, I think that's his real hair. He's in several videos, but it's a group of chemists at this university in Great Britain. I've forgotten which
university in Nottingham.
And they have put together a series of videos about all of the elements. So you can choose an element in the table, and there'll be a little video about it. You know, you can look at them adding lithium to water, or doing other cool, visually interesting.
Excuse me.
Did you click one?
Yeah.
Oops. Right. Oh, yes.
Okay. No problem.
And they're on YouTube as well, but I went ahead and linked to their main site. And it's just a really cool, I think, really cool introduction to some of the more interesting chemistry that's out there that doesn't require working out balanced equations. It's just
this is great.
It's great stuff.
I mean, as an educational tool.
Yeah,
it's nice.
If you're a kid in taking your first chemistry course or something like that, what could be more boring than the periodic table?
Right.
And this is a great way to give it real meaning. This is fantastic.
Nice. It's very nice. This gives me an idea. Each of us who work on a virus can make a short movie about that virus,
and put it together in a classification scheme.
And you have some kind, not the periodic table, but some kind of
maybe a classification.
Maybe a classification.
Thing,
click on a virus and you get Rich Condit. 3 minutes talking about vaccine, and people can submit their videos, and you can have more than one for each virus. You can get different people's viewpoints, right?
That's a good idea.
Yeah, it is a good idea.
To think about how to do that. We could put a couple of up ourselves to sort of seed the jar, and then see if it takes off. That would be fun. That is neat to show it to my son. See if he was always saying, why do I have to memorize the periodic chart? I can always find it online. But, of course, it helps you understand what's going on if you memorize it, right?
Sure.
Doesn't seem to get it. My pick is something I discovered or was re-alerted to this week. I was preparing my lecture on virus structure, and I was over at the Protein Data Bank, which is the repository for all the coordinates of proteins, all proteins whose structures have been solved. And they actually have a very nice section called Educational Resources, where you can get posters, calendars, tutorials, animations, a variety of lessons that they've put together, exhibit materials, just a bunch of good stuff about protein structure.
Oh, neat.
One of my favorites is this big poster. It's called The Quick Reference Guide to Molecules in Living Cells. It's down there about halfway down under posters. It's a beautiful poster, quite big, which has a variety of molecules and viruses on it, so you get a sense of scale. So this is good stuff. It's good teaching stuff, and I highly recommend you check it out. It's the PDB Educational Resources page. Now, the PDB is full of coordinates, which may not interest most people, but this particular page, which we'll link to, is very nice.
I'm just trying to get the poster up here. I'm a little slow.
You click on that.
Yeah.
Should download it as a PDF.
Yeah, it's my My machine's just slow. That's all.
It'll come down while we're wrapping up here.
Right. Okay.
Well, that'll do it for another TWiV, and you ought to digest this one slowly because we won't have one for another 2 weeks. I have study section next week, and that's going to consume me pretty much for the whole week. I have to go to NIH and review grants to decide which one or two will get money from the government. And so I'll get some more ideas for TWiV there, for no doubt, and possibly corral some people into joining us in the future. So come back in 2 weeks, and we'll be back. If you're a new listener, go to iTunes and leave a comment if you can. That really helps us to stay on the front page, so more and more people will find us. As always, send your comments and questions to twiv@twiv.tv. We love to get them. TWiV is part of sciencepodcasters.org and promednetwork.com, as well as microbeworld.org, which are all websites where you can find other science resources, including podcasts from a wide variety of fields. Hey, Rich, thanks for joining us again.
Well, thanks for having me. I really enjoyed it.
I think
Good fun.
You'll be back probably in 2 weeks, right?
Yep.
Maybe we'll have a
2 or 3 weeks. I forget what the calendar looks like. But yes, I'll be back as soon as I can.
And hopefully Dick will be back. And Rich, of course, is at the University of Florida at Gainesville, where it's probably 70 degrees and sunny at the moment.
71 and
Amazing.
A little bit overcast, but not bad.
Alan, dovedocs.com.
Yes.
Thank you.
Always a pleasure.
Pretty soon you're having an article in Nature Medicine. Is that right?
Yes, it's a profile of a researcher, and she was very interesting to talk to, and I hope the profile gives a good impression of her.
How soon do we expect that to come out?
That is slated for the March issue.
It'll be a while.
I will plug it on the show when it.
All right. Well, thanks for joining us again. Thanks, everyone, for listening. You've been listening, of course, to This Week in Virology. As far as I know, the only podcast dedicated to viruses. Who else would do it? Right? Thanks for joining us, everyone. We'll be back in 2 weeks. Another TWiV is viral.